Hello AP Biology students, let's go over the 14th section in Unit 6. And in this section we're going to talk about something called DNA fingerprinting. DNA fingerprinting is going to use a type of biotechnology called gel electrophoresis. This is just a laboratory method of separating mixtures of DNA, RNA, or proteins. These machines will separate these molecules based on their molecular size. Typically, the smaller particles or molecules travel further than the larger ones. See, in our gel plate at the top, we have a mixture of macromolecules that's separated based on length, but the smaller ones traveling further than the larger ones. In DNA fingerprinting, DNA is going to be cut by something called a restriction enzyme. These fragments of DNA then are going to be added to wells in the plate. Think of the wells here as just being like little indentations where the macromolecules are going to start. An electrical current is then going to separate the DNA based on size because DNA is negative. The smaller fragments of DNA are going to travel further than the larger fragments do. That's again because DNA is negative and the DNA is going to travel towards the positive end. Now when the bands are done traveling, they're going to leave lines in the gel that we can read. Like I said before, the smallest fragment is going to travel the furthest, while the largest fragments aren't going to travel very far away from the well. We typically see more DNA leaving a thicker band, while less DNA leaving a thinner band. Now what we're actually separating are things called STRs and VNTRs. These are short repeated sequences of DNA between around 2 and 9 bases that are going to repeat several times randomly for different people. The variation we see in these STRs and VNTRs allows us to statistically say whether or not a sample of DNA matches somebody. Now the VNTRs are just going to be a little bit longer. They're about 10 to 100 bases, but they are again repeats of DNA that typically don't produce proteins. This method of separating DNA and identifying somebody's DNA, maybe at like a crime scene, was first discovered in 1984. And it's been a major tool in forensics, and it's slowly being replaced by genome sequencing, but it's still actively being used in the field. This is a lot cheaper of a method of identifying somebody compared with genome sequencing, so it's definitely used by forensics. But basically what they're looking at are these STRs and VNTRs that are different between each person. As you can see here, we have 3 different people, and at each location they have a different set or different number of these repeats. So when we compare suspect's DNA to one that was found at a crime scene, we can say that it was statistically implausible that it's anyone else. We can also tell if a child belongs to a mother or father because typically half of the bands or half of these STRs and VNTRs come from each parent. So you can see here our child has half the bands from the mother and half the bands from the father. If they have a band that doesn't come from the mother, we know it has to come from the father.